Welcome to the third video in the topic of biological molecules. This one is all about carbohydrates, and they are simply what the word says they are. They are hydrates of carbon. Hydrates of carbon, something like this. Carbon, hydrate means water. I'll just put that like that. That's their empirical formula. That's the simplest version of a carbohydrate you can get. Now, in this video, we're going to be pulling bits of information from the previous two videos. So if you want to go back and have a look at those to refresh your memory, that's fine. But here, we're going to start with this as an empirical formula. And empirical formula is just the simplest version of, so if you take the subscripts here and you reduce them all down, that's the simplest molecular formula for a carbohydrate. That's what that term empirical formula means. What you've already done in science is you learned about photosynthesis and glucose and all that probably many times over, and you probably recall that glucose is C6H12O6, glucose. Well, if you look at that, you divide that by 6, that's what you're going to get, isn't it? It's a carbohydrate. Now, glucose, product of photosynthesis, is fundamental to this topic. We could represent it this way as a chemical formula, but glucose quite often when it's produced, it's actually a linear molecule. It's more like this. If I can fit it in here. 6 carbons. I'm going to show you how all the pieces fit in here now. You see, and what I had in the previous video was that carbon has 4 bonds, hydrogen has 1 bond, and oxygen has 2 bonds. And if you go through what I'm just drawing here and pick all those pieces out, you'll see it works. This is glucose as a linear molecule. Here's an oxygen, bonded once here. It's also bonded to the hydrogen, although I don't draw the bond in there. There is a bond connecting those two. Hydrogen bonded to oxygen, hydrogen bonded to oxygen, and this oxygen's bonded in 2 ways. Every carbon here has 4 bonds around it. Even this one down here with the aldehyde group on it, double bond O on the end of a molecule, that's an aldehyde group. There's 1, 2, 3, 4 bonds on that carbon, 1 on that hydrogen, 2 on that oxygen. That's what this carbohydrate called glucose looks like when it's being made, or when it's made by photosynthesis. We typically don't draw it that way. Typically we draw it because in a cyclic fashion. And what happens to form the cyclic version is this molecule is actually big enough that it can bend around on itself, so that this part here, I'm just going to be careful I get this in the right spot, this part here can bond around onto this part. So there's the cyclic version of it here. And then the hydrogen atoms reorient themselves. I'll draw the cyclic version the way it's typically represented over here. So we have carbon, carbon, carbon in the cyclic fashion now, carbon. This carbon is this carbon here. This CH2OH is up here. CHOH, right? So that's that part up there. And what's missing in here is this oxygen is in here. Now I haven't drawn all the hydrogens and hydroxides on, so I'll just put those in here like this. There's the hydroxides, the way they exist in this molecule, and the hydrogens. So when you look at glucose as a molecule, we need 1 more bond in here. We need another hydrogen. As glucose as a molecule, C6H12O6, that's typically what 1 molecule of glucose looks like. Carbons. Let's just go through and count, make sure I didn't miss any. C6H12O6 is what I've got to find here. 1, 2, 3, 4, 5, 6 carbons. Oxygen. 1, 2, 3, 4, 5, 6 oxygens. 12 carbons. 1, 2, 3. 4, 5, 6, 7, 8, 9, 10, 11. I miscounted. Must have miscounted. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12. They're all there. So there's glucose. That's what glucose as the simple carbohydrate looks like. Product of photosynthesis. Most common sugar. Sugars end in OSE. And as soon as I say that, you're going to say, oh yeah, I've heard of other molecules that are, they end in OSE. Well, yeah, there's other molecules which are sugar molecules. That's where this topic's going to go. So if you remember the last video, we mentioned something like dehydration synthesis, and where we took little molecules, we combined them together to get bigger molecules. Well, that's what I'm going to represent here right now. I'm going to take glucose, I'll just use this space. I'm going to use that molecule as well. I'm going to use this space right here to refresh our memories here. So if we took 2 unit molecules of glucose and we did dehydration synthesis, we're going to wind up with a bigger molecule that's made out of 2 parts. So this is glucose plus glucose. We get water given off. This molecule here, this is maltose. Glucose plus glucose is maltose. Now, let's just consider this for a second. Glucose is C6H12O6. What do you think the formula for maltose is? 2 glucoses together, right? Minus a molecule of water. C6H12, no, C12, because we've got 2 of them. H, it should be 24, but it's only 22 because we took out 2 for that O11. That's maltose. Now I can show you, I left that on here so I can show you what that looks like using chemical formulas when they're drawn out like that. So if we take that as 1 molecule of glucose and we put the next molecule of glucose right next to it here. So what I'm doing is I'm drawing that portion, I'm going to draw the molecule going across. So we've got a carbon here, we've got oxygen here, carbon here, carbon here, carbon here, carbon here. We've got the CH2OH up here, and we've got the hydrogen, hydroxide, hydrogen, hydroxide, hydrogen, hydroxide. There's the second molecule of glucose, and we're going to combine them together. So this combines with this. I think you can see where the water comes from. It comes right here. Right here, look. This is the water molecule. And the bond from sugar to sugar, from this glucose molecule to this glucose molecule, is actually a thing called an ester bond. The bond goes from carbon to oxygen to carbon. So it looks like this. Oops. C, oxygen, back to carbon. That's this carbon here. That carbon's that one. That oxygen is right there. That's an ester bond. So that's just one specific example. Just erase this so it's clearer. Just one specific example of how you can take a simple sugar like glucose, combine it together using dehydration synthesis, which is a topic we've already covered, and you wind up with a double sugar like maltose. These things, simple sugars, are called monosaccharides. Monosaccharide, it just means sugar. This one, it's a double sugar, it's a disaccharide. Double sugar. There's lots of examples of these things, and I'll erase this here because we don't need it right now, and I'll show you some of these examples of other sugar molecules. And you get used to these names because, the OSE ending and so on. We have glucose, which we just mentioned, we know that one. There's also fructose. OSE endings, remember. This is fruit sugar. It's also C6H12O6. We could have galactose. It's another one. It's also C6H12O6. And you start to wonder, well, how can you have all these molecules with exact same chemical formulas? Well, the answer is that they're not exactly put together the same way. They've got the same parts in total, but they're attached slightly differently. We know what glucose looks like as a molecule. Fructose is not the same. This is because it's a six-sided sugar, like this, with the oxygen there. Fructose is actually not a hexose like this. Hexose, which is a six-sided sugar. This is actually a pentose, which means it's a five-sided sugar. And you know, diagrammatically, the oxygen that's in the molecule is up here, for example. So we have a molecule that's more that shape than that shape. And galactose is a hexose sugar. So those are different monosaccharides. There's also two that are going to show up in another video, the last video in this series, ribose and deoxyribose. And you're going to be thinking, wait a minute, I think I know deoxyribose because we talk about DNA, deoxyribonucleic acid. Deoxyribonucleic acid, DNA, has this as a sugar in it. That's where that name comes from. Again, notice the OSE endings. These are all monosaccharides. Well, there's a bunch of disaccharides too. This is one common example, but if I take this, I'm going to use the picture here, and I don't want to use glucose plus glucose, I won't get maltose all the time. If I took glucose plus galactose, I'll wind up with a disaccharide known as lactose. And you think, wait a minute, lactose. Lactose intolerance. Lactose is milk sugar. Milk sugar. That's where that comes from. Or if I wanted to take glucose and fructose, and put them together, I'm not going to get lactose. What I get instead is sucrose, which is ordinary table sugar. So that's the thing that we get with carbohydrates. We know what the simple sugars are like, but you know, it can keep going. You don't have to just get a disaccharide, because if you think back to that molecule, both sides had a carbon. Just take me a second here to sketch this again. Both sides have both sides have got a hydrogen hydroxide on it, and the ester bond formed between two sugars in this fashion. But if you think about it for a split second, you'll realize that it doesn't have to stop there. If the bonding can occur here, bonding could also occur here on another one, or it could occur over here on another one, and it could keep going. You could get longer and longer and longer, not trisaccharides so much, but we call them oligosaccharides. Now, oligo is a prefix that means few. So they have a few sugar molecules in them. Monosaccharide, disaccharide is two, oligo is a few. Well, how many is a few? Well, really, it's between 3 and 9. Once you get more than 9, it becomes a fairly large molecule, and it's called a polysaccharide. Just want to sit back to the functions of some of these things. I've sort of glossed over that part already. Glucose, primary energy molecule. That's its function. It's made by photosynthesis. We ingest glucose. Glucose is burned in our cells, and that's how we get ATP energy. Sucrose, lactose, galactose, sucrose, lactose, maltose, all those double sugars, all those disaccharides, those are sort of like storage, temporary storage. We don't really keep them in our body in that form for very long, but they are there, and we can use it to make glucose by hydrolysis very quickly and access the ATP from there. Once they get into this length of a molecule, they start to have other functions. And I'll just mention some of that here. Just for example, oligosaccharides are antigens on blood cells, for example. So if you think about a blood cell, a biconcave blood cell, it's got little sugar chains hanging off its surface. These are oligosaccharides. And there's different ones, like there's ones which are, there's only like 6 carbons long, but that's the difference between type A blood and type B blood and type AB blood, is exactly which oligosaccharides they have on them. So that's another function for sugar molecules. This topic comes up in another video in the future. We'll get to that one. So that's, they do have other functions. Polysaccharides, this is just long-term storage of energy for us. I mean, if we get a bunch of sugar and we wind up with too much sugar in our body, then in our blood takes it and it stores it in our liver in the form of glycogen. If plants have more sugar than they need, they're generating glucose by photosynthesis all the time. What they do with it, it goes into their sap and it goes down to their roots for storage, or into starch tissues for storage. So we have glycogen and starch as storage molecules. There is a third polysaccharide, and I'll mention that one right now, but then I want to draw some comparisons in just a second. So there's primarily three polysaccharides. They're very important to know: starch, glycogen, and cellulose. Cellulose is not a long-term storage molecule. Cellulose is actually the carbohydrate that makes up cell walls in plants. And that's the topic I want to end this video with, but I want to do it this way. We have polysaccharides. One of the things I capitalize on every chance I get is contrasts between things. Like if you know a structure, you know a substance, or you know some topic, can you make a contrast between that topic or that structure or that substance and another one? Well, that's a good example this is a good example of how to do that. And I'll say it oftentimes that if you can contrast these things, we have a structure you can contrast, you have a function you can contrast, and you have a location that can be contrasted. Structure, function, location. Oftentimes things in biology can be contrasted in terms of structure, function, and location. So I said there's three polysaccharides. We've got starch, glycogen, and cellulose. Structure. How is starch different from glycogen? Well, they're both polysaccharides. They're both polyglucose. In fact, all three of these things are polyglucose. So that's something that's in common with their structure. But how are they different in terms of contrast? This one is slightly branched and has a little spirally parts on it. Now, if you think about the structure of glucose, there's OHs sticking out all over that molecule. Well, it doesn't have to just bond end to end to end to end. It can actually bond end and then maybe on this side a little bit. So you get, little branches formed. So glucose is starch is slightly branched, and some of the times it actually forms little spiral pieces. Glycogen is highly branched. Cellulose is simply linear. It doesn't branch. These have the same kind of, remember I said ester bonds have got the bond, goes from carbon to oxygen back to carbon. These ones have got the same ester bonds. These ones have a different ester bond. In fact, pattern. In fact, what happens with this one, in these ones, polymers of glucose, starch, or glucose, in a line, and in a line, and sometimes it branches. Cellulose takes the glucose and takes the next glucose molecule is actually inverted compared to the first one. So the ester bond, the bonding pattern is totally different. We can digest starch. We can digest glycogen. We cannot digest cellulose. This is dietary fiber in us. So in terms of function, this is energy storage. It's made by plants, but we can use it. It's a long-term energy storage molecule. Same thing here. This is in our liver. Oops, that's the location. In liver tissue. This is in plant tissue. Cellulose function is to form cell walls. In us, it's dietary fiber. We cannot digest it. We eat it, it forms part of our feces. And location here, plant cell walls. Function, so this is a strengthening molecule. Strength in plant cell walls. Okay. So I think that sort of wraps up all this business about carbohydrates. You get a pretty good idea of what they're like. The next video is about lipids, and they're quite different as well. Thank you very much for watching. Remember, if you want to subscribe to my videos, remember the little green gecko over here. That's how you subscribe. Click on that. Thanks very much.